Hi guys, so in this video we're going to go over B1.2 proteins. This is both the SL and the HL content, but the HL only content will be posted at a later time. So yeah, let's get into it. Proteins are made up of amino acids, okay? Amino acids are the polymer for the protein or polypeptide polymer. Keep in mind, proteins and polypeptides can be used interchangeably. They're more or less synonyms of each other. But yeah, so amino acids are what proteins are made of, and I've drawn here the general formula. Key things to note is that we have the central carbon, we have a carboxyl group, we have an amino group, or amine, and we have this R group. Now this R group, we don't really care about it right now. It's just another side chain that, different amino acids have different components, obviously depending on the amino acid, but these are the main ones that we need to care about, which are the amine and the carboxyl group, which every amino acid has, okay? Now these amino acids, okay, we know that they're the monomers, but how do they become proteins? Well, they undergo something called condensation reactions, and that's when a peptide bond forms between two amino acids to add it onto the polypeptide chain. So with 2 amino acids, it's dipeptide. 3 amino acids or more, it's a polypeptide. And you can kind of infer this from their prefixes di and poly. Now, in a condensation reaction, we always get a main product and a side product, okay? And the side product is always smaller, like molecularly, like the physical molecule is smaller than the main molecule. And in this case, the main molecule is a polypeptide, I've just abbreviated here, and the side one is water, okay? Now let me draw out a condensation reaction for you guys. We have Now, this OH and the H over here are going to form water together, and a link between these two, or the peptide bond, forms, creating this dipeptide. So if we draw out the final structure, we find that there's this thing made, and that is what you call the peptide bond. Okay? Now, in our body, there are 20 amino acids, and they can be sorted in 3 different ways. So, in our body, there are 20 amino acids that can be used by ribosomes to make polypeptides, okay, and synthesized by ribosomes. So, basically, they're essential, non-essential, or conditionally essential, okay? And the key difference between these is if they can or cannot be synthesized by humans, okay? So I'm only speaking in the context of humans. Anyway, so 9 of the 20 amino acids that ribosomes use are called essential amino acids, and that is because they cannot be synthesized by humans. They cannot be synthesized by us, okay? Now, the other 11 are either non-essential or conditionally essential. Non-essential means they can be synthesized, and conditionally essential, this is interesting. So basically, that means they normally, if everything's normal, they can be synthesized by the human body, but if, in times of illness or stress, they cannot be synthesized. So, yeah. Now, okay, if 9 of these 20 cannot be synthesized by us, where do we get them from? If ribosomes need them, where do we get them from? Well, we consume them. We get them from our diets, okay? Now, one thing that the IB wants us to know is that vegan diets, particularly, may not always get these sources because of the restrictions they have, or for that matter, anyone else on a serious diet, or for whatever reason. So they require close attention to ensure that all amino acids are consumed, and so a varied diet is important, so it could include these sources. It's a kind of random point to know, but yeah. Anyway, with these 20 amino acids rises a lot of variety, okay? So amino acids or polypeptides there's almost like an infinite possibility of the type of polypeptide chain that can form, and that's because any polypeptide can use any of the 20 amino acids. They can be arranged in any order, they can be any length, and also some polypeptides are modified after synthesis, adding another layer of variation, okay? We don't really need to know about the modification process in this topic, but it's important to know what happens. Now, one way to like work out the number of possibilities is this little equation. You don't always need to use it. I don't think you ever really need to use it, but it helps for your understanding. So N in this case stands for the number of amino acids being used. So and 20 is obviously the type of amino acid. So if we have a polypeptide chain of 5, it'd be 20 to the power of 5, and that's how many possibilities of polypeptides there are. Okay. For this spec point, you do need to know varied examples of polypeptides, but I'll leave that up to your exploration because, you can pick whichever one you want. You can use popular ones that come up in the IB spec itself, like enzymes like lipase, or hormones like insulin, or even hemoglobin. So just find out about them, their purposes, and what. Now we're going to go over protein functionality, and one thing to keep note is that protein function depends on its structure, its 3D structure specifically. Let me add that in, okay? And this is because of the intermolecular bonds between the molecules, and they are weak, and they can be disrupted, and that causes the changes in formation and shape, okay? So weak intermolecular forces allow for change in structure, and this is obviously not what we want, and a change in structure is called denaturation. Okay, so when an enzyme or protein gets denatured, that means its 3D structure has broken and it doesn't return to its former structure. And there are two main causes for this, either changes in temperature or changes in pH. Okay, now I'm going to speak in terms of enzymes, but this can be applied to all proteins, okay? It's just enzymes that are an easier example. So if we look at temperature, the graph for enzyme activity would look something like this, okay? This would be temp, this would be enzyme activity, okay? Now what happens is if we follow this graph, temperature increases, and enzyme activity increases because of, more energy, more collisions, more whatnot, and it reaches its optimum point. That is its highest point. That's when enzyme activity is the most effective, whatnot, okay? But after that temperature, okay, there's a sudden drop, and that's because the enzymes have denatured, or the proteins have denatured. So now they don't function normally because the shape has changed. And one thing you know about enzymes is that they have an active site that allows for reactions to take place. And if that active site changes shape, it cannot work and is therefore denatured. Okay, so what happens is heat is past its optimum, and then hydrogen bonds are overcome. Okay, now the next factor that impacts structure is pH levels. Okay, now the graph looks slightly different. It's more of a bell curve. Again, this would be enzyme activity, this would be pH level. Okay, and over here is the optimum. And in this case, unless the enzyme isn't constantly looking at this pH, it will be denatured. Anything too low, anything too high, it's too acidic or too basic, okay? And that's because of the ions. So, in an amino acid, the amino group, so the NH2 group, serves as, it's basic, it accepts protons, okay? And then the carboxyl group is acidic because it donates protons. So in this case, if it's too high for the optimal or too low, it will prevent normal hydrogen bonding because there are extra ions, either H plus or OH. If it's too acidic, there are more H plus ions. If it's too basic, there are more OH minus ions, which disrupt the bonding for COOH and NH2, thus causing a change in shape.